Good morning, guys. So we're going to be starting our unit on genetics, chromosomal genetics, which is where you're dealing with Punnett squares and things like that. But before we start, there's just kind of some review that we need to go over and an introduction of some information that you'll need to help you work through genetics problems in AP. In AP, we don't do too many Punnett squares. In other words, there's a shortcut. You don't have to draw out Punnett squares. However, if you don't like the shortcut, you can always draw out a Punnett square. So before we start talking about that, this unit is over genetics and meiosis. So just real quickly, I want to talk about a couple points of meiosis before we get into the details that you'll need to know. Meiosis is similar to mitosis in that it's a type of cell division. However, in meiosis, the point is to not create two cells that are identical like you do in mitosis. In meiosis, we're creating gametes. Gametes are sperm and egg. And we do that, or before that happens, we have to cut the chromosome number in half. That's because at some point a sperm will fertilize an egg, and that will bring the number of chromosomes back up to full. So for example, humans have 46 chromosomes. Those chromosomes are in pairs. You have 23 pairs. The reason that you have pairs is that you got one chromosome number 1 from mom, one chromosome number 1 from dad, one chromosome number 13 from mom, one from dad. So we have our chromosomes in pairs. The reason that I wanted to mention meiosis up front, meiosis is very important in introducing variation in the population. So variation we talked about during evolution, and we understand from an evolutionary standpoint that variation is very critical in a population. So where does that variation come from? We discussed mutation. Mutation is kind of the root of all variation, but a lot of variation can come about during sexual reproduction. And the reason is because we're taking two organisms and combining their genetics to give us some variation. Variation comes from three different events that occur during meiosis and sexual reproduction. One is independent assortment. We'll talk about that in more detail, but that's basically where during meiosis, as a cell splits up its chromosomes, cuts the chromosome number in half, which of the pair goes into the new cell is completely random. So chromosome pairs in meiosis line up at the equator of the cell and then separate. And either the paternal or the maternal chromosome will go into one cell and the other one will go into another cell. So that kind of shuffles up our genes. We'll go into that in more detail later. A second place where variation can occur is during crossing over. Crossing over occurs in meiosis as well, and it occurs when the pairing up of homologous chromosomes. For example, chromosome number 13, let's say. You've got one from mom, one from dad. Those chromosomes are homologous. They share the same traits. They might have different versions of those traits, but they do have the same traits. So crossing over occurs when those chromosomes get really close to one another and kind of exchange some information. And again, that's something we'll get into detail with later too. A third place where we get variation is in random fertilization. And what this means is that whichever sperm is created due to meiosis, what sperm fertilizes what egg is random. So you can get all kinds of possibilities in a given sperm and all kinds of possibilities in a given egg, and how those possibilities end up together creates a lot of variation. That's something we'll talk about later as well. I just kind of wanted to introduce that to you right up front. Now, I don't know if you're familiar with genetics problems, but I'm just going to give you a couple of examples of Mendelian genetics. And what I mean by Mendelian genetics is it's very basic genetics where you have a dominant allele and a recessive allele. So there's a couple that I want to point out. So let's take this problem first. Let's say, and I'm not even going to identify the trait. We're just going to say that there's an individual who is heterozygous for a given trait A. Heterozygous means they have a dominant and a recessive allele. And we're going to cross that individual with another heterozygous individual. If you do the Punnett square for that, hopefully you remember how to do all this. You can see that we end up with 3 individuals who have the dominant trait and 1 individual who has the recessive trait. So that ratio is 3-to-1 dominant to recessive. Another example that I want us to look at is if we take a heterozygous individual and cross it with a homozygous recessive individual. If you do that Punnett square, you can see that these two are dominant and these two are recessive. So it's a 2-to-2 or 1-to-1 ratio. For every dominant, there's a recessive. I need you to memorize these two ratios and know that if you see a 3-to-1 ratio in your data, and you'll see what I'm talking about with your assignment today. We're going to look at genetics of corn. If you see a 3-to-1 ratio in your data, you know that there are 3 dominant to 1 recessive. You also know that these are the parents. That's always the ratio that you get with those parents. By the same token, whenever you have a 1-to-1 ratio, so you have roughly equal number of the dominant and recessive trait, that will give you a 1-to-1 ratio. These are your parents. So in other words, memorize this ratio, and then you don't have to work out those genetics problems anymore. They work themselves out. You already know what they are. Now there's 2 more ratios that I want to show you, and I'm not going to work an entire problem because you'll see why in just a minute. But these are double trait problems or dihybrid problems. And the first one that I want to kind of show you is if I take an individual who is heterozygous for 2 traits. All right, so trait A and trait B, they're heterozygous. They have a dominant and a recessive allele. And the same thing for the other individual. Now, if you were to work out this Punnett square, it's going to have 16 boxes, which is why I'm not going to work it out. And I will not necessarily make you guys work it out either. But the way that you would set this up, when you set up a Punnett square, what you're showing are the possible offspring from this cross. What you're putting here, like we did here, so we had this cross here, these are the possible offspring from this cross. What this A is what the gamete could be. So this parent could contribute a big A, or they could contribute a little a. So these are the gametes. So how do you get the gametes if you've got double traits? Well, each gamete's got to have an A and a B. So you just you can use a distributive thing like you would in math, or you can just kind of figure it out. You got to have an A and a B, both of them big. You're going to have a little a and a little b. You're going to have a big A combined with a little b, and you're going to have a little a combined with the big B. And you'll have the same thing here. And you can go ahead and do this problem. And honestly, I recommend that you do it once so that you can see where these ratios are coming from. But if you do this cross, what you will find is this ratio, 9 to 3 to 3 to 1, where 9 have both dominant traits. The 1 is where your individual has both recessive traits. This 3 would be where they have one dominant, let's say the A, and then one recessive, let's say the B. And then this one would be where they have one recessive the other way. So a little A, and one dominant. Say that would be the B. And you'll see that if you do the Punnett square. Now, what I want you to memorize so that you don't have to do these Punnett squares is this ratio here. If you have two heterozygous parents crossed with one another, that's your ratio. The last ratio that I want you to memorize is another two-trait ratio. And that's if we have a heterozygous parent crossed with a double homozygous recessive parent. This is called a test cross. And this is done if you know the phenotype or what this individual looks like. You know that they exhibit both dominant traits. But when you see a dominant trait, you don't know. Think back to Hardy Weinberg. If it's dominant, you don't know if this individual is big A, or big A, little a. You can't tell just by seeing a dominant trait. So what a test cross does is allow us to see if this is a heterozygous or homozygous individual. And we'll kind of talk about that a little bit more later. Bottom line, what I want you to remember from this cross is the ratio that you get. And do the Punnett square for that so that you can see where that ratio comes from. It's actually much shorter than this one because if we need to get all the gametes possible here, you have big A, big B, big A, little b, little a, big B, and little a, little b. So that's what I can get from, those are the possible gametes for that parent. The possible gametes for this parent are a little easier. It's just A and B. There's nothing else that you can get. So you really just have to do one row. And what you'll find out is that you'll have a 1 to 1 to 1 to 1 ratio. And your 1 will be both dominant, both recessive, a dominant and a recessive dominant. And you'll kind of see that if you do the Punnett square. All right, these ratios that I'm asking you to memorize, these 4 ratios, so the 9 to 3 to 3 to 1, the 1 to 1 to 1 to 1, the 3 to 1, and the 1 to 1. These will help you do your chi-square analysis because you're going to be looking at actual data. You'll read through the lab and you'll see what I'm talking about. What you're going to be doing in your chi-square is to see if your null hypothesis that the ratio is 3 to 1 because the parents are big A, little a, and big A, little a, that these two things match. But you're going to be looking at data like you're going to have 75 to 25. Is that a 3 to 1 ratio? And you'll do a chi-square to figure that out. So you'll need to know this will be your expected or what you think you should get if the parents are heterozygous, which is a 3 to 1 ratio. See if you can figure it out. I'll post a video tomorrow or later today that shows you how to do that.